Greetings, class, and welcome back to the last part of our chapter. In this particular part of the chapter, we're going to be concentrating on some different variations on Mendel's law, some things that are a little bit different than what we learned about in the previous part of the chapter, because not everything that Mendel discovered with his humble pea plants actually explains everything about genetics. So we do have some special cases that we're going to call as beyond basic crosses. So we're going to look at some slightly different rule applications for some of the genetic things that we see. Well, first we'll start with something called incomplete dominance. Incomplete dominance is just like it sounds. What we've been looking at before was something called complete dominance, where we had one completely dominant allele and we had a recessive allele, which was always masked in the presence of the dominant. But that's not how it always works. Okay, sometimes when you combine a dominant with a recessive, you end up with something in between. Okay, you kind of mix them together and you end up with an intermediary phenotype. Okay, that's what incomplete dominance. It's like crossing a tall and a short and not ending up with tall like Mendel had discovered, but ending up with medium height. Okay, so we call that incomplete dominance. And it looks something like this. We have neither allele is completely dominant. Okay, so to have said dominant recessive is not 100% accurate, but sometimes you will see it annotated by a capital and a lowercase, and I'll show you a different way in a second. And in the case of the capital and the lowercase, we do see the capital codes for the red, the lowercase codes for the white. So in both the homozygous forms, we see one particular extreme or the other, either the red or the white. But when we combine them together and we end up with a heterozygote, what we end up with is not either red or white, we end up with something in between. So I guess there really is no true dominant or recessive in this case, okay, but they blend their traits together when they combined. Okay, so this is called incomplete dominance. It's also notated this way, where we have like a base letter to indicate, the basic trait we're looking at, the character, and then a superscript to indicate the alleles, in this case, capital R and capital W. So it's notated slightly different here, so you can see there's no lowercase here. If we have R and R, we end up with red. If we have W and W, we end up with white. But if we end up with a heterozygous form, we don't end up with red or white, we end up with pink. And it also looks something like this and can be demonstrated with a Punnett square. So like I said, sometimes you'll see the lowercase r like as if it were recessive, but that's just the way they notate it. There's really not a true recessive or dominant in this case. Okay? But sometimes you'll see it like I showed you on the previous slide. Sometimes you have something called codominance, and codominance sounds the same as incomplete dominance, but it's not. So in the case of codominance, you literally have two dominants, whereas in the other one, it's not really a true recessive, not really a true dominant, some kind of quasi kind of thing. But in this case, we literally have two dominant traits, and each one is so powerful that they don't blend together and create an intermediary. They both have to actually be expressed. So in the case of this chicken, you cross a chicken with white feathers with a chicken with the black feathers. If it was incomplete dominance, we would end up with a chicken with gray feathers. But in this case, we end up with black and white feathers, something they called an erminate chicken. Okay, so you can see both are actually exhibited. Blood is also another example. We have type A blood and we have type B blood, but we also have type AB blood because type A and type B are both codominant. Here's some other examples. You can see the flowers on the left. Purple and white doesn't give you purple or white or a light color, intermediary color like in incomplete dominance. You literally end up with purple and white specks together. In a roan cow, crossing a cow with red fur and a cow with white fur doesn't give you like a light red or an orangish rustish kind of thing. You literally see red and white spots because both alleles are dominant, so both are going to be presented in the phenotype. And crossing looks quite similar. The Punnett squares basically function the same. If we have homozygous versions, we'll see one of the phenotypes, but in the heterozygote, we see both of them. So in this case, RW for all of them, so they're all roan. Sometimes we have special cases where we have multiple alleles, where there's actually more than two alleles for a trait. Like everything we've learned so far was either one or the other. So like for example, in blood, we have three different alleles. We have A, B, and we have type O, which is the recessive version. So that's actually indicated with the lowercase I. Okay. So when the lowercase I or type O is combined with either A or B, it will serve as the recessive. And then you would either have, in case of type A, we could be homozygous type A or we could be heterozygous type A. O is recessive to A or B. Okay. But when A and B are together, they're codominant. So we have a lot of things going on with blood typing. But again, you still only get 2 alleles, but there's more than 2 to choose from. Sometimes you have some special cases where one gene can actually influence multiple characters. We call that pleiotropy, and an example of that would be sickle cell anemia, which we'll actually talk a little bit more about in the next chapter. That's a disease in which the red blood cells take on this kind of odd sickle shape, and it can be quite dangerous because what happens is the sickle cells, as I'll show you in the next slide, actually can clog up the arteries and the blood vessels, so to speak, as they get hooked on different areas of the blood vessels, which I'll show you. But what's kind of interesting is carriers of sickle cell disease actually have a resistance to malaria. So there's kind of an evolutionary adaptation in there as well. But those that actually exhibit the disorder, it can be quite painful. This is a sickle cell here, so you can imagine they don't store the oxygen very well, so you have increased cases of anemia. That's why they call it sickle cell anemia, because you're not able to deliver the blood efficiently and carry it around the body. But also the way that this is shaped at these points here, sometimes they have a tendency to get caught in the bends and the turns of the blood vessels, and then one gets stuck with another and stuck with another, and then next thing you know, blood flow is being blocked. So there could be some mild symptoms, but then there also could be some much more dangerous symptoms depending on the severity of the case. Sometimes we have what's called polygenic inheritance. Polygenic inheritance is when a single phenotype is the result of multiple genes. That's what poly means, more than one gene interact together. So now we're not talking about multiple alleles here. In that case, we're talking about several alleles for one gene. But now we're talking about multiple genes working together for a phenotype. Human height is an example of one. Skin color, eye color, these are examples. In this case here, look at this Punnett square here. You can see there are 3 different genes working there. You can see 2 pairs or a pair of alleles for 3 different traits or 3 different genes there. So how they all intersect together will be the phenotype you'll see. So it does make quite a much bigger Punnett square there. There's 64 different possible cases that can occur there when you have 3 different genes working together to exhibit a phenotype. Something else that can affect the actual expression of your genes in terms of what you see as the phenotype can be environmental factors. So it's not only 100% genetics. Some characters are also, the phenotype is also a result of the interaction with the environment. So heredity has part to do with it, and so does environment. Remember this one from the test, the Himalayan rabbit there, who changes color based on the temperature around them. So like if that Himalayan rabbit stays in South Florida, living in the warmth, it's going to be white furred. But if it gets in cold conditions, remember like this, its fur can actually get much darker. So that's just one example. We could have people that are predisposed based on heredity to be a certain height or a certain body type, and then maybe perhaps they live in poverty and they're malnourished and the environment is not favorable for them to actually achieve their genetic potential. So the environment does have an impact. So for the last part of the chapter now, I want to concentrate on the chromosomal basis of inheritance, which we've kind of already talked about already a little bit, and then finish up with sex-linked traits. So the chromosomal theory of inheritance holds 2 things. First, that genes occupy specific loci or positions on the chromosomes, which we already learned about, right? You can find a particular gene on one chromosome and in the same place on the homologous chromosome. You'll find the code might be slightly different, but it's coding for the same character. And chromosomes will undergo segregation and independent assortment during meiosis. Remember, they'll line up right to left or left to right, whatever, and then they'll separate from each other so that only one chromosome gets passed on. And a lot of this understanding of chromosomes and how they work was due to Mendel's work. And so we can demonstrate that, of course, with the dihybrid crosses, which we looked at before. Remember, we could have independent assortment here. We could have the capital R on the left, the little r on the right, or we could have little r on the right, big R on the left. And each one of those, the way they line up, will create a different outcome once we get to the gametes on the bottom. If you look at them now, the gametes are different. Okay, so it's 2 equal probable arrangements, like one or the other. And then from there, remember, they'll segregate out, and so that's how we'll end up with what we end up with in the gametes. And this is, of course, what the Punnett square would look like to represent the different possible possibilities. Now, one of the things that scientists have also discovered, which is a little bit different than what Mendel learned, but because we talked about in an earlier chapter, when we talked about independent assortment, how we learned some things are not necessarily attached to each other. When we were looking at more than one trait, like when we were doing the dihybrid crosses, But when William Bates and Reginald Punnett did work with plants, they noticed that some genes were linked together. So the patterns of behavior, depending on the type of organism, was different. Okay, so they didn't necessarily get the 9 to 3 to 3 to 1 ratio that Mendel found when he worked with his pea plants. So they've kind of determined that some genes do travel together, and it depends on their location. How close or far apart they are will determine whether or not they'll travel together or become separated. So like, for example, here, crossing this type of flower and this type of pollen, if you went based on Mendel's work, you would expect these kinds of numbers. But surprisingly, this is the kind of numbers they got. Look at the purple and long. They tended to really stay together much longer, which led them to kind of believe that perhaps their proximity was very close together on the chromosome. And then notice some of these others, much less likely to be together. So maybe they were further from each other. Now, some of this, of course, has to do with crossing over, remember, which we talked about. Some linked genes can be separated by crossing over, and some may not, depending on how close they are. Maybe they separate together, or maybe they stay together when the crossing over occurs. But remember, we call that crossing over and the rearrangement of the genes as they're transferred from one chromosome to the other. We call those, the resulting gametes, recombinant gametes, because their genetic information has been recombined together. And the percentage of recombinant offspring amongst the total is called recombination frequency. And we're not going to talk too much about this in this introductory bio. Of course, if you take AP bio later, you'll learn more about that. But by looking at the frequencies of how different alleles travel with each other, different characters stayed together or separated, that kind of stuff, scientists are able to kind of put things together and figure out where things are in relationship to each other. Okay. And so what they're able to do by looking at things like, for example, the tendency of particular traits to stay together or the tendency for them to recombine in different ways, it allows geneticists and scientists to create what's called a genetic map. And a genetic map is an ordered list of genetic loci along a chromosome, like the placement of this particular character, this particular character, this particular character. Okay, and a genetic map that also indicates the recombinant frequencies or the chances that they will stay together, travel apart from each other, those kind of things, is called a linkage map. And this is basically what a genetic map looks like here, and you can see where the particular traits are in terms of their order. Like you can see the eye color, the antennae, the wings, those kind of things, and in the order or loci that they would be found in. So like on the homologous pair, you might have a different form of the character, but they're in the same basic spot. And by studying how these things recombine with each other and how they show up in offspring and successive generations, scientists were able to kind of put this thing together without actually physically being able to see their positions on the chromosomes at the time. So the last thing I want to talk about are sex chromosomes and sex-linked genes. These are a little bit different in how they behave. Okay, because remember, what we're talking about here is the 23rd pair of chromosomes. Remember, the other 22 is what we've been talking about so far, which are the autosomes, or the non-sex chromosomes. The 23rd pair is, remember, the X and Y chromosomes. Remember, males are XY and females are XX. Okay, and they're, of course, not really literally XY. The Y doesn't really look like a Y. It's more of this. There's more of a size difference, but because of the difference in their shape and size and stuff, there's pieces of information that's missing on the Y that appears on the X. So it changes the pattern of behavior of how phenotypes are expressed and how alleles are passed on. Now, some organisms are affected by the presence of the X and Y chromosomes. The Y has genes for the development, say, of the testes in males, and the absence of the Y allows for the female structures to develop. But some species, like some certain types of reptiles, are affected by temperature. Depending on the temperature will ultimately determine the gender of the offspring. But how do the sex-linked chromosomes, the genes themselves, how are they affected by the structure of the actual chromosome? So what we're going to concentrate on primarily are sex-linked traits that are caused by genes on the X chromosome. Okay, so unless otherwise you're told, we'll assume these are on the X chromosome. And sex-linked traits tend to be recessive, but they're not all. Okay, and so what happens is with these sex-linked traits, it's kind of interesting, is generally females are not usually afflicted with these types of disorders, or we don't see the expressions of these types of traits in them because they have two X chromosomes. And so they actually have to receive two alleles, especially it's recessive. Males have only one X chromosome, so they don't have the second homologous chromosome to actually mask or hide the allele. So whatever allele they get on the X chromosome will always be expressed. If it's dominant or recessive, that will be expressed in the male. And the male, their expression of whatever that chromosome is, they'll receive that from their mother because their mother will pass that on to them. Two examples that we'll look at are color blindness and hemophilia. Color blindness, of course, is in your ability to discern certain colors. Hemophilia is your inability to clot your blood properly. Okay? But this is why it happens. Okay, so above here we have a female. You can see she has brown eyes. She's heterozygous. She has the capital B here and the little b here. So the brown masks the blue, and so she ends up with brown eyes. However, in the case of the male, he has the blue-eyed allele, but he does not have, because the chromosome is smaller and different in shape, he does not have the dominant brown eyes, so there's nothing to mask it, so he only needs one allele. Okay? For the female to have exhibited blue eyes, she would have to receive the blue allele from both her mom and her dad. Okay, so in this case, you can see the difference and why certain types of traits or phenotypes are more often exhibited in males than females. And again, here, this is an actual look. So it doesn't really look like a Y, but look at the size difference. I mean, look, it's missing. I mean, it's just not the same size and shape as the X. So there are things that are going to be missing on the Y that you find on the X. Now, a gene that is linked on a sex chromosome is called a sex-linked gene. X chromosomes, the genes that are found on those are called X-linked genes. And sometimes we'll see an X-linked gene, or sometimes we'll see a Y-linked gene. And there are a couple of the cases, like we'll look at in some of our practice, like ear hair, that's found on the Y chromosome. But any gene that's located on either of the sex chromosomes is called a sex-linked gene. We're going to concentrate primarily on those found on the X chromosome for this chapter, but you may have one or two that are on the Y chromosome. Okay. The fruit flies, like we looked at in the video we watched the other day with Thomas Hunt Morgan, showed, he kind of made this discovery when he noticed there was uneven trends in how he was seeing the eye color being transferred to the male and female fruit flies. Okay. So this is what we're talking about, the eye color here. Okay. Now, red is the dominant and white is the recessive. So in the case if you make a cross between parent, one that's white and one that's red, the offspring will end up red because the female has both dominant alleles and she passes the X on to the male, so the male will be red. The male passes on this one, but since the female is dominant, she masks it. So this female is called a carrier. Since it's heterozygous, it exhibits the dominant phenotype, but it carries the recessive allele and it can pass it on. Here's a different example. In this case here, you see the female does carry one of the recessive alleles. So in this case here, there is a potential for her to actually pass on white offspring, but you would only see it in the male, because the father has the dominant R and he'll pass it on to the females. So regardless of what combination they get, they'll have at least one dominant R. Okay, so but for the female to actually exhibit the white eyes, she would have to get the recessive from her mom and her dad. So the dad would have to exhibit the white eyes, and the mom would have to either be white-eyed or heterozygous. So the chances of that are very slim. Okay. So you can see it, though it does happen once in a while. Like here we have an example of a female that is a heterozygote. She's a carrier, but she has red eyes, and a male that does exhibit white eyes. And so you can see there is a potential cross where they intersect together, and we do end up with a white-eyed female. Now, you do have to be careful when you're answering these types of questions to see what they're asking. If they're asking in terms of the offspring or the gender of the offspring. And I'm going to go back a slide. So what do I mean by that? So let's say, for example, I have this cross of a heterozygous mom and dominant, what they call normal male. If you have both dominant alleles, we say that we're normal. Okay, so the female is heterozygous, she's a carrier, and the male is normal for red-eyedness. So what is the chance of having white-eyed offspring? Okay, so a simple question like that, 1 out of 4, right? So it's 1 out of 4, 25%. But what if I change the question? I said, what is the chance of having a male that has white eyes? Now, it's 1 out of 2, right? So it would be 50%. What if I said, what is the chance of having a female that also has white eyes? 0 out of 4, right? But what's the chance of having a red-eyed female? The females, there's 2 of them. Both of them are red-eyed, right? So it would be 2 out of 2, or 100%. So you just have to listen to or read the question carefully, see what it's actually asking. As I mentioned already, most X-linked human disorders are caused by recessive alleles, and so consequently they're most often seen in males because the male receives a single X-linked recessive allele from his mother, and he gets nothing from his father to block it out, but a female has to receive the allele from both parents. And we can demonstrate that, of course, in the Punnett square and in the pedigree. Okay, so in this case here, we have a pedigree of the European royal families. In this particular case, this is the Romanov Russian royal family, which date all the way back to Queen Victoria of England, who had a daughter named Alice, who had a daughter named Alexandra. And you can see these three were all carriers. Okay, and Alexandra actually passed on color blindness to her son Alexis. Okay? But you'll notice in a much bigger blowout of a pedigree here, this is actually the royal families much further back. This is what we were just looking at here. But notice the prevalence of all the males that exhibit it, all these squares that are complete shaded in are males. Notice there are no carriers that are male. Male cannot be carriers of an X-linked trait because they only have one allele, so they'll exhibit it, either the dominant or recessive version. But notice the females will be carriers. Notice all the female carriers. I'm circling here. So there's a ton. But you notice, and historically they've never found or recorded any females that actually exhibited hemophilia in this line. I mean, there are some they don't necessarily know about. There's some question marks here because they didn't have offspring, so they can't tell for sure. But you'll notice there are just so many males because, again, the male only needs to have one allele. The females have to have two. And so that's the end of our chapter. Don't forget to look over the study guide questions here at the back end, the things you should be able to do. And, of course, there's a lot of great practice and reviews at the top of the module as well.